A very good morning, dear student. Today I will teach you the microorganisms. It would be the second part, and in this microorganism topic, you know the role of microorganism in our daily life. The advantages of microorganism is certainly there, and as well as there are some harmful effects of microbes in our daily life. So, before taking this topic, this module, I would like to begin from the first thing, that is the importance of bacteria, microbes in our life. This is very important topic, and in this topic you will see there are various role of bacteria, virus, protozoa, fungi and algae are there. So in the microorganisms role, very first we should see about the important role of microorganisms, the advantages of microorganisms. Many topic you have heard, you have studied so far, like in the last class you have studied a very important bacteria which is increasing the soil fertility. The first which increase the soil fertility, and that is also a very good example of mutualism, and that is rhizobium. Rhizobium bacteria is present in the pulse root. You have notices like root nodules of the pulses contains a special bacteria that is called rhizobium, and rhizobium bacteria is fixing the atmospheric nitrogen in the soil, and by this they are increasing the fertility of soil, right? So when we are going to start the important role of microorganisms or advantages of microbes, we have to write. Number one, you will begin with this, the soil fertility. The soil fertility. As I just told you, in the root nodules, the nitrogen fixing bacteria rhizobium bacteria is present in the root nodules of the pulse family, and what is the special thing in this, that these nitrogen fixing bacteria is full of one gene, special gene, and that is NIF gene. For your information, NIF gene means Ni for nitrogen, so got my point. NIF gene, nitrogen fixing genes. Look at this, and these are present in the root nodules of the pulse family. Any pulses, they are having root nodules, and in that root nodules one is special bacteria that is fixing the nitrogen in the soil, and that is NIF gene, nitrogen fixing genes. Okay, so as far as the important role of microorganism is concerned, I am moving to the next one, and that is for making of curd and bread. You have heard some bacteria name, you could remember your past knowledge, that the second one is the making of curd and bread. So in the bakery one special bacteria which is playing important role, and that is the lactobacillus. The lactobacillus bacteria is converting the milk into the curd, and that is why it is known as lactobacillus. So you can write in the bakery industry the lactobacillus is playing important role. So you have to write the name of this bacteria and the name of this bacteria which converts the milk into curd, that is called lactobacillus. So you have to write this lactobacillus. After this, there is another one big role in the industry, in the bakery industry, and somewhere you can see the which is used for making cakes and pastries, and by which process it is going to be completed, and I am going to indicate that is fermentation process. Number 3, some bacteria are performing the fermentation process, like in the making wine. The fermentation process takes place. Sugar is converted into alcohol by the yeast. So this is known as the fermentation process. Yeast reproduces rapidly and produces CO2 during respiration. And in this, the duff bubbles of gas fills the duff and increase the volume of. This is the basis of use of yeast in the baking industry, in the bakery, making bread, pastries and cakes. So fermentation process in which we are preparing the wine, we are preparing the alcohol, and in this the sugar is converted into alcohol, and because of the presence of the yeast. So another role of microorganism in the fermentation process for making pastries and cakes. Now moving to the next one, and which one is the number 4, the commercial use. You have heard the commercial uses of microorganism. Like just I told you, the conversion in the large scale, when we convert the sugar into alcohol, it is known as fermentation process, and it is commercial use. By this process we can use the commercial use, and the fermentation process we can make many more things like the pastries and these cakes. So for the next one, number 4, you have heard some role of bacteria in the medicine industry. So I'm going to write the medicinal use. Okay. So in medicinal use, many medicines are prepared from the bacteria and fungi, but one special things I would like to mention, that in the medicine industry, the antibiotics. Have you heard this name? Yes. Many of you have heard. For the treatment, in the pathology, for the treatment, we should always support with the antibiotics. So what are the antibiotics? Antibiotics are prepared from the microorganisms and is used to stop the growth of microorganism. They provide resistance power, fighting power to the body. There are some natural antibiotics like colostrum. In the mother milk it is there, but passive immunity for the outside of the body, there are some antibiotics which is given, and from where it is prepared? The antibiotics are prepared from the microorganism, and first antibiotics is discovered by Alexander Fleming, and the example of the first antibiotics is penicillin, right? So you can write antibiotics. Example of this, the first antibiotic is the penicillin, and it is discovered by the Alexander Fleming. You should remember the name of Alexander Fleming. So when we start the medicinal use of microbes, we must remember the role of antibiotics, which is prepared from the microbes and used to stop the growth of microorganisms. And the first antibiotic is the penicillin, discovered by the scientist Alexander Fleming. Now, you must remember the cleaning the environment. Yes, there are decomposer which eat dead and decay matter, like many bacteria and fungi, which are saprophytes, which eat dead and decay matter. You will surprised to know that after death of animals and plants, they are going to be decomposed in the soil. That is why this earth is cleaned. So this saprophytic process, the earth is clean. So you can write cleaning the environment. The fifth use, you can write cleaning the we'll write the example. Decomposer. So when you start the important role of microbes, you have to write the first one is soil fertility. As nitrogen fixing bacteria, rhizobium is playing important role for the nitrogen fixation in the soil, and that bacteria is present in the pulse root. Number two, making of curd and bread by the lactobacillus bacteria. The milk is converted into curd. And the third one is for the commercial use of microbes. The fermentation process takes place through which we can prepare the alcohol, we can prepare the pastries, we can prepare the cakes. By this fermentation, many eatables are going to be prepared in the fermentation process, by this fermentation process. As I told you, the medicinal use, the big industry is there, the medicine, the antibiotics, prepared from the microorganism, and to stop the growth of microorganisms, the first antibiotic is prepared from the Alexander Fleming, right? So when we are moving the important role, the advantages of microbes, these are the advantages. And opposite to this, when we are going to study the harmful effect of the microorganism, before that, one thing I would like to tell you, one important thing, that you have heard the vaccination process. In the vaccination, like entire universe is in the search of vaccine. And what is special things vaccine? How does this vaccination process play important role, and how does this vaccine is first time discovered? You have heard one scientist named Edward Jenner, and this vaccine term is originated from the Latin word, and that is vacca, which means cow. The vacca means cow. Cowpox vaccine discovered the smallpox vaccine. Then there are series of the vaccine, as you know, BCG vaccine, polio vaccine, ATS vaccine, DPT vaccine. There are many hepatitis vaccine which prevent the liver infection. So there are series of the vaccine in our country, but what we do in the vaccination process, right? So when we are going to study the role of microbes, we must see there is a very wide role of microbes in the vaccination process. How does these microbes work in the vaccination? So first, vaccination. Vaccination is the process in which we introduce the vaccine in the body, right? Preventing infection. This is the method of preventing infection in the body. In the vaccination, what we do, we take vaccine, and this vaccine is different from the medicine. That medicine affect after the disease, but this vaccination is having a special quality. In this process, vaccine recognize the enemy and it stops the infection and kills that pathogen. This vaccination is a method of preventing infection from the body. This vaccination, in this process, vaccine introduced in the body which prevents the infection in the body, which prevent the infection. Now, how it can prevent? What is special quality in this? Okay, you know, vaccine always started working before the disease, and it stopped the growth of bacteria. You will be surprised to know that if it is going to kill the bacteria, why does not affect our body? Our body could also damage from there could be a little bit damage in our body. No, there would not be any damage to the body, because these vaccines are introduced in our body in the inactive form, in the sleeping stage. They don't act when there is no need of action. Until unless there will be attack of that microbes, these vaccine will remain silent phase, will remain in the inactive phase, and they will not work. But at the time of infection, at the time of injury, at the time of attack, when any pathogens invade, when any attack is there in the body, at that time they do two things. Vaccines, first, recognition of their enemy, and second thing, after recognition, after diagnosis, they kills that enemy. So this is the great thing in the vaccination. That is why all over the entire universe, entire country is in the search of coronavirus. Until unless the vaccine would not be developed, there would not be complete control of this virus. That is why the scientists are in the search of vaccine. There are two quality in the vaccine. Number one, they diagnose the pathogen, and then they kills that pathogen and they stop the growth of pathogen. So vaccination is a process in which we are putting vaccine in the body, and vaccine, what is the special quality? They are having a memory cell, and memory cell, they recognize, they memorizes the particular bacteria, virus, and then they stop. Because you have heard that there are some vaccine, examples of vaccine. You know the vaccine, polio vaccine. Now you will be getting any patient of polio. Then you must be knowing the BCG vaccine, BCG, Bacillus calmette-guerin, anti-tuberculosis, anti-TB you can write, anti-tuberculosis, then anti-tetanus, TET vaccine, ATS vaccine you can, ATS vaccine, anti-tetanus serum. Anti-tetanus serum, this vaccine is preventing the tetanus bacteria attack. So particular vaccine like hepatitis vaccine. Hepatitis. What is meaning of hepatica? Hepatica is the name of liver. So to stop the liver infections, to stop the disease of liver, we always go for the hepatitis vaccine. Hepatitis A, hepatitis B. You have heard so many vaccines. So these are few examples of the vaccine, right? So when you are going to study vaccination process, what we do in the vaccination, who discovered the first vaccine? Edward Jenner. Edward Jenner discovered the first vaccine. Okay, process of introducing the vaccine. Edward Jenner discovered the first this vaccine, and you know the smallpox vaccine, cowpox vaccine is introduced, and there are series of the vaccine in this. Now, it was about the role and important role of microbes in our life. But opposite to this, when we are going, there are different role of microbes, there are useful, important role, there are advantages of microbes, and simply there are some disadvantages, harmful effect of microbes. So you must remember, there is animal disease, there are plant disease. So you have to see, when we are going for the study, you must remember few words. When we are going for study, to study the disease, as I told you in my first module, in the first lecture, that there are some disease. So what we should remember, for the study of disease, we should remember the pathogen, name of pathogen, then symptoms, and then prevention and control. So in the pathology, as I told you, in the pathology, first thing, pathogen. What are the pathogen? Disease causing microorganisms. So you must remember, disease causing microbes are called pathogen. Which causes the disease, these are called pathogen. So before beginning any disease, in matter of disease, what we should remember? We should remember the name of pathogen. So when we start the table, so in table you should remember the name of disease, name of pathogen, symptoms, mode of prevention, name of disease, pathogen, mode of transmission, mode of transmission. So if I am going to write, so you have to remember the name, like malaria. What is the malaria? The name of pathogen is a protozoa. You can write Plasmodium vivax. Plasmodium vivax, it is a type of protozoa. It is a protozoa which is present in the female Anopheles mosquito, and the mode of transmission is the body. You can write, although there is no need of writing the contact. Through contact they can reach from one person to another. They are not communicable, they are non-communicable. Okay, so when you are going to start the mode of transmission, through the contact, air, water and soil. So it is not very tough. So you can write only two columns, name of disease and the name of pathogen first. Second, you have heard the typhoid. Which is this? In the typhoid, these are the bacterial disease, and typhoid is caused by a bacteria, Mycobacterium typhi. So in short form you can write MTYPHAE is a bacteria. Microbacterium typhi is the causal organism or pathogen. Then you know the tuberculosis. Tuberculosis, or in short form you can write TB. This tuberculosis, which is very dangerous disease, and it is also a type of bacterial disease, and this is caused by the M. tuberculi, like M. microbacterium. So M. tuberculi, M, I can write Mycobacterium tuberculi, right? So malaria, typhoid, tuberculosis, and then you can write the cholera. Also, this is also a bacterial disease. M. cholerae, M. cholerae. So it is not very tough to understand, tough to learn the name. M for microbacterium. It stands for microbacterium. So Mycobacterium typhi for the typhoid, the bacteria tuberculi for the TB, and the third one is Mycobacterium cholerae, which is causing the cholera, the extra loss of water and minerals in the body. All of a sudden, when loose motions and vomiting started, this happens. Is to be called as an epidemic 30-40 years back, but now it is controlled after the development of the vaccine. There are many diseases, you know, the AIDS. It's a type of virus, HIV-3 virus. Okay, so AIDS is called occurred by the one pathogen that is called virus, HIV virus. So when you start, you have to write the table also for these. So at the end, I would like to go for the some harmful effect of the microbes also, like you have to mention the disease, some bacterial disease, some viral disease, some fungal disease in the plants and in animal. And in the last I would like to give few assignment question for you. In this you have to write the number 1, define vaccine and pathogen. Mention any 4 advantages of microbes. Mention any 4 main role of bacteria, or you can three advantages of microbes in our daily life. Now you can also explain the vaccination process with the example. Explain the vaccination process. Explain the vaccination process with the examples. With the example. And number 4, make a table of some bacterial and viral disease. Make a table of bacterial and table of bacterial and viral. Bacterial and so in this you have to write the name of disease, and opposite to that you have to write the pathogen name. Okay, and the last one you can write what is antibiotic. Very important. What is what is antibiotics? So you have to write all these 5 assignment questions, and in this you have studied. Once again I would like to go for recapitulation that what are the advantages, what are the disadvantages, what diseases are there, and you have to complete these 4 assignment questions. Okay, so for today's lecture from my side, I am sure that after the completion of this module you will be able to write the answer of these 5 questions. Okay, and I would be coming with the next module, and that would be the role of bacteria in the food preservation, the role of microbes, what are the methods of food preservation, in the next lecture. Thank you.